Hello and welcome to this session, LO3, Interpreting Data on Factors Affecting Enzyme Activity. Today, we will explore how temperature and pH influence enzyme activity. This session focuses on understanding how changes in conditions affect enzyme function and how data interpretation helps reveal patterns in enzyme behavior. A temperature versus reaction rate graph often shows a gradual increase initially. The peak represents the optimum temperature for enzyme activity. Beyond this, the reaction rate declines sharply. This is due to the breaking of bonds stabilizing the enzyme's structure. If the rate continues to rise, the optimum temperature hasn't been exceeded. A bell-shaped graph illustrates pH versus reaction rate. The peak indicates the optimum pH for enzyme activity. Deviations cause a decrease in activity due to altered hydrogen bonding. Extreme pH values can lead to enzyme denaturation. Recognizing these patterns helps identify enzyme function in different environments. Initially, substrate concentration increases reaction rate rapidly. As all active sites become occupied, the rate levels off. This plateau is known as Vmax. If no plateau is observed, substrate concentration might still be limiting. Understanding this helps in identifying enzyme saturation. An increase in enzyme concentration typically raises reaction rate. This is true if substrate is in excess. The graph shows a straight line through the origin. If the rate levels off, substrate becomes the limiting factor. This illustrates the relationship between enzyme concentration and activity. Competitive inhibition shows reduced reaction rate at low substrate concentration. The same Vmax is achieved eventually with more substrate. This indicates the inhibitor competes with the substrate at the active site. Understanding this helps in identifying competitive inhibition. It alters substrate binding efficiency. Non-competitive inhibition results in a lower maximum reaction rate. Increasing substrate concentration does not restore the original maximum. This suggests the inhibitor binds elsewhere on the enzyme. It alters the enzyme's shape, affecting its function. Recognizing these patterns aids in identifying non-competitive inhibition. Denaturation is evident when reaction rate drops sharply without recovery. Enzyme activity remains low even if conditions normalize. This indicates irreversible loss of function. Recognizing denaturation is crucial for understanding enzyme stability. It highlights the importance of maintaining optimal conditions. When interpreting enzyme data, identify the independent variable. Observe the trend in reaction rate. Link these changes to enzyme structure and function. Determine if inhibition, saturation, or denaturation has occurred. This approach helps in making informed conclusions about enzyme activity. Temperature affects enzyme function by altering kinetic energy. Within a moderate range, reaction rate doubles for every 10 degrees increase. Each enzyme has an optimum temperature. Beyond this, bonds break, leading to denaturation. This understanding is crucial for controlling enzyme activity. Exceeding optimum temperature breaks stabilizing bonds. The enzyme's shape changes, distorting the active site. Substrate binding reduces, leading to denaturation. This process results in loss of catalytic function. Recognizing this helps in maintaining enzyme stability. pH influences ionic and hydrogen bonding in enzymes. Each enzyme has an optimum pH for peak function. Deviations alter the charge of amino acid side chains. This disrupts bonding, changing the active site's shape. Extreme pH can lead to irreversible denaturation. Enzymes adapt to specific pH conditions in the body. Pepsin functions optimally in the stomach's acidic environment. Salivary amylase works best at neutral pH. Trypsin operates efficiently in the slightly alkaline small intestine. These adaptations ensure maximum enzyme efficiency. Temperature and pH affect enzyme activity by altering structure. Moderate temperature increases reaction rate through kinetic energy. Excessive heat causes denaturation. Optimal pH maintains bonding and charge in the active site. Extreme pH conditions reduce catalytic efficiency. Interpreting enzyme data involves linking graphical trends to molecular explanations. Rising temperature increases collisions until denaturation. pH influences bonding in the active site. Increasing substrate concentration leads to saturation and Vmax. Competitive and non-competitive inhibition affect catalytic capacity differently. Temperature affects the kinetic energy of enzyme and substrate molecules. Molecules move faster, increasing collision frequency. More enzyme-substrate complexes form, raising reaction rate. The temperature coefficient, Q10, describes this relationship. Each enzyme has an optimum temperature for peak activity. Extreme pH changes the charge of amino acids. This disrupts ionic bonds and modifies hydrogen bonding. The active site's shape changes, reducing substrate binding. As a result, reaction rate decreases significantly. Recognizing these changes is crucial for enzyme stability. Enzymes in the gastrointestinal tract adapt to specific pH conditions. Pepsin operates in acidic environments like the stomach. Salivary amylase functions best at neutral pH in the mouth. Trypsin excels in the slightly alkaline small intestine. These adaptations optimize enzyme efficiency. Temperature and pH influence enzyme activity by altering structure. Moderate temperature increases reaction rate due to higher kinetic energy. Excessive heat causes denaturation, reducing function. Optimal pH maintains correct bonding in the active site. Extreme pH alters structure and reduces efficiency. Understanding inhibition and denaturation is crucial. This knowledge is vital for biological and industrial applications. Should you have any questions, please contact us via WhatsApp at 074-843-61688. Our team will gladly support you. Thanks for your time, and see you soon.